here, and today instead of having two talks, we're going to have the luxury of watching one. Our guest today is Jessica Goodheart, all the way from New York, and it's always a treat for us to have somebody from across the pond. So Jessica is at the American Museum of Natural History, and she is an assistant curator there in malacology and an assistant professor in invertebrate zoology at the Richard Gilder Graduate School, if I'm correct. Now, I think that Jessica would not be insulted if I would label her a malacologist with a particular love for nudibranchs, but she's worked on a variety of invertebrates, including ostracods and flatworms, and even things with backbones such as salamanders. I think she works mostly with phylogenetic methods, but she studies a range of topics, including bioluminescence. But today, we've invited her to talk about something that is dear to our hearts, which is to do with toxins and venoms. And she's going to talk about how and why some predatory creatures eat cnidarians, and especially their venom delivery organs and what they do with them. So Jessica, thank you for joining us this afternoon. The digital podium is yours.
Thank you very much. Let me go ahead and share my screen here. Don't know where that ended up. There it is. Okay. I assume you can see that okay?
Yep.
Great. Okay, well, thank you for the invitation. I'm excited to talk to you all today. I'm really curious about what this particular audience thinks of my work. I don't necessarily study the venoms that the organisms I'm interested in steal, but really curious about the kinds of questions that people who are interested in venoms might think of with regard to the system. But before I get into the details of my work and kind of present to you some of my more recent research on this topic, I first want to take a little bit of a step back and help sort of put into place the kinds of questions I'm interested in, because they're generally much more broad evolutionary questions than just thinking about nematocyst sequestration, which is what I study. So like many biologists, I'm interested in diversity. I think this is pretty common for a lot of us. And I'm interested in understanding essentially how evolution shapes the diversity of form and function we see in extant organisms. Most of my work has been in metazoans, so that's often what I think about. But I think a lot of these kinds of interesting questions can be applicable to understanding organisms at a variety of scales. And when I think about understanding biodiversity and sort of the complexity of life, one of the ways I think about it is that often we focus on sort of a genome or an organism where they're sort of evolving along a lineage, and its genes are changing, the sort of phenotypes are changing within this organism in order to create diversity. But we don't necessarily always think about the fact that a lot of these phenotypes that are evolving within organisms not necessarily interact with the environment around them, but rely on the environment around them in order to function. And so often I think about this in terms of theft, where organisms are actually stealing chemicals, structures, et cetera, from other organisms in order to generate the phenotypes that exist. And so when we think about theft, it's actually quite common in nature and has multiple functions. So there's two main functions that generally theft is involved in. The first is metabolism. So thinking about things like cnidarian dinoflagellate endosymbiosis, or chloroplast sequestration or kleptoplasty that we actually see in some sea slugs, but also some other organisms like forams, for example. And then we have another function or other kind of functions that go together, which are in defense and prey capture. And so this can be anywhere from a boxer crab, hopefully you can see my pointer, a boxer crab here that attaches anemones to its claws, down to specific chemicals that are sequestered in, say, arthropod or sea slug lineages for defense. And then in other cases, we have like this midshipman fish down here in the bottom right that actually steals bioluminescent chemicals from its ostracod prey in order to produce counter shading as a means of sort of protection. And kind of in between these two more extremes is the sequestration of structures. So these are things like nematocysts or the organelles from cnidarians, which I'll talk a bit about in a second, where it's in some ways less complex than thinking about the sequestration of dinoflagellates or chloroplasts, because it doesn't necessarily come with its own genome, but the structure itself, the organelle, is much more complex than thinking about a specific chemical being sequestered. So it's a kind of happy medium. And then on top of that, nematocysts, which I'll sort of go through in a second for those of you who aren't familiar, are the only defensive organelle that are stolen and stored intracellularly, at least as far as I'm aware. So if anyone knows of any others, I'd certainly be happy to hear about it. But for those of you that aren't familiar, nematocysts are these organelles that are found usually lining the epithelium of cnidarians, like this Exaptasia anemone here on the left. If we look at what those sort of generally look like inside the organism, nematocysts are the organelle. They're generated or constructed by these special cells called nematocytes. And the nematocyst itself is made up of a capsule and an reversible shaft and or tubule, depending on the type of nematocyst, that when fired, as you can see on the right here, when these are fired, they are sort of inject a complement of venoms or a mix of venoms into the potential predator or prey item as a means of protecting themselves or capturing their prey. And these venoms can be a mix depending on the nematocyst type, because there's different types depending on what specific organism you're looking at. There's different types depending on which tissue you're looking at. So it's a really diverse system when thinking about nematocysts. And so when we try to understand how nematocysts, or when we think about nematocysts in terms of their cnidarian function, it's really interesting that organisms are able to steal these, because the intention is that these are meant to be defensive, right? And so not only are these organisms able to protect themselves from these structures, but then they're able to steal them and maintain their function. And this is what I'm interested in understanding, how this happens and how this evolved. And I study this in nudibranchs. Nudibranchs are gastropods, marine gastropods that are shellless as adults. So they have a shell as a larva, and then during metamorphosis, they lose it. And many researchers have hypothesized that this loss of a shell has actually led to sort of diversification of alternative defense mechanisms. So this can include things like their bright color patterns, which may be aposematic in terms of sort of trying to deter predators that way. Some of them are actually cryptic rather than aposematic. And then they're also able to steal things like stealing chemicals from their prey items, and then also nematocysts. So with all of this theft that's happening within this group, it's a really interesting group in terms of understanding nematocyst sequestration, in terms of understanding sequestration more broadly, but also thinking about nematocyst sequestration or theft in particular, because it's the most diverse group in which this has evolved. There's around 600 or so species that do this in nudibranchs. So it just provides for a nice system. I want to show you exactly kind of what this looks like in terms of morphology before I get into the details. This is a nudibranch. The top right here is the head of the animal. When the animal eats the cnidarian, they break up the tissue. The nematocysts, the organelle, get separated from their cells during this process. And nematocysts are then passed through their digestive system, which branches out into these sort of finger-like projections off of the dorsum here called serrata. And if we look more closely at one of these here, inside each of these serrata is this branching of the digestive gland. You can see it in sort of this brownish orange color here. Nematocysts move up through that and into the very distal tip of the serous into a structure that we call the cnidosac. And this is where these organelles are stolen and stored for long-term use. And each one of these serrata has a cnidosac in it. And if we were to look inside of that cnidosac and try and see what exactly is happening, this is a histological section of the tip of a cerus from this animal, Dentice occidentalis, here on the left. The tip or distal end of the cerus is at the top. The proximal end is at the bottom. Down here is digestive gland. And if we look, there's this little path here from the digestive gland, which is into the cnidosac, which is outlined by these darker blue muscle striations here. And inside the cnidosac, we actually find these specialized cells. Hopefully you can see the outline here. And these cells are called cnidophages. This is where nematocysts, which are shown here in these kind of darker blue oblong dots. And that's where nematocysts are sort of sequestered intracellularly and stored for the long term. So when we think about this system, it's really important to understand not just that nematocysts are sequestered within cnidophages, but how exactly this happens, because we only know that it does. We don't necessarily know what the sort of actual mechanisms are of this particular phenotype. For example, how nematocysts are targeted or how they maintain their function. And when we think about trying to understand this, we have to sort of try and figure out what potential processes they might be using. And phagocytosis is the most likely. So for those of you who don't, haven't thought about it in a while, phagocytosis is the process of cells engulfing and internalizing large cells or particles. So these are just generally larger cells or particles. Often you can think of like viruses, bacteria. These sorts of immune cells or phagocytes do often function in things like immunity. And the way this usually works is the virus, for example, is identified via a complement of receptors. Often these phagocytes are generalists, so they'll just sort of target a lot of different things. They'll internalize the virus into a phagosome. The phagosome then gets merged with heterolysosomes for digestion, and then the sort of extra particles that aren't used are exocytosed. That's kind of generally the process of what's happening with phagocytosis and intracellular digestion. So the question then becomes, how exactly did the cells that sequester nematocysts evolve to target them? Are there specific receptors or other types of mechanisms that allow these cells to exclusively take up nematocysts over other structures that are present in their prey? And to answer these questions about sort of where and how nematocysts are selected, we needed a laboratory system. So I'm going to focus on that in particular today and talk to you about what I've done in the past few years. And I'm happy to answer questions about this system or any of the other parts of my work as well. So to do that, we are in our lab, we're using the emerging model nudibranch called Bergia stephaniae. This is something I started working on in my previous postdoc, and I brought with me to my lab at AMNH. Some benefits of this species, it's actually a local US species, technically local. It's from the Florida Keys, so it's a tropical species. The nice things are we have a reproducible laboratory and staging system, and part of that is because this particular species doesn't seem to need external cues to actually metamorphose, which is unusual among nudibranchs. It also feeds on another easy to culture anemone species, Exaptasia diaphana. And the reason this is particularly important is that Exaptasia is actually considered a pest in the aquarium trade, and so they're actually extremely easy to culture because most people don't want to, and somehow they still end up there. I want to show you what it looks like when the Bergia feed, because I always think that's a fun thing to see. The anemone here, the Exaptasia anemone is in the middle, and you'll see the Bergia kind of come around and they'll do what we call a mobbing behavior, essentially, to sort of all attack the anemone at once. And this is a means to protect themselves, most likely, because the anemones can eat the nudibranchs as well, and will, if they're big enough and the nudibranch is small enough. And they're able to eat this animal all together within about an hour and a half. This is, I hope it's obvious, a sped up version of that, because they are actually quite slow, despite being voracious predators. And then finally, I've been working on a chromosome level genome paper that will soon be available, hopefully. We're currently in revisions, but the preprint is on bioRxiv if anyone is curious. So we have this system that we're able to use to try and investigate sequestration or this theft of nematocysts. And so if we want to answer this question about where and how selectivity is occurring, we first had to come up with some hypotheses as to, where this might be happening. And if we think about sequestration, so this is the digestive gland, it's sort of a cartoon of the digestive gland down here into the knapsack structure. And in the digestive gland, there's a lot of different types of tissues. In Aptasia, we might see dinoflagellates, spirocysts, in addition to nematocysts. And so the question becomes, where is selectivity actually happening? Where are they only picking out nematocysts? And there's sort of two general hypothesized sites of selectivity that came up for us immediately. The first was with the nidophages themselves, so that they're able to, again, have receptors that target nematocysts in particular. But a second one was that at this entrance to the digestive gland, there's a sphincter. And it was possible that there's some other sort of protective mechanism that essentially only allows for nematocysts to enter the cnidosac, in which case nidophages would not necessarily need to have any receptors specific to nematocysts. They might just take up anything that's there inside of the cnidosac. And to try and investigate this, adults are very difficult to see the cnidosac and what's happening inside of the animal because they have sort of structural color that makes it difficult. So we've actually been using Berghia juveniles because they sequester nematocysts very early on in development while they're still quite translucent. And so we're able to actually image them in ways that we can't do so well with the adults. And I want to show you a bit about what this looks like so you know what the structures look like in these animals. This first image is a pre-feeding juvenile. So this is post-metamorphosis, but it has not yet fed on any anemones. The colors are acetylated alpha-tubulin in pink, so it's cilia, things like that. Nematocysts would be in cyan, but there aren't any because it hasn't fed yet. And then phalloidin is in yellow. So that's basically staining the musculature in these animals. And I'm hoping you'll notice there's no cyan, there's no nematocyst. And if we look at the sort of mantle margins, we actually don't see that there's any cnidosac or sign of any cnidosac structure at this very early stage. So pre-feeding, they don't have any cnidosacs, they don't sequester nematocysts. Within 2 to 4 days after they eat their first anemone, they're already sequestering nematocysts. So this is extremely fast in the world of slugs, I suppose. Where you can already see these sort of blue or cyan clusters of nematocysts here at the mantle margins. And if we zoom in on one of these, there's a yellow outline surrounding those, indicating that there's a muscular structure that's already present, indicating that the cnidosacs have developed, maybe not necessarily to the full capacity as an adult, but the structure is there and functional at this very early stage. And so if we want to see what's happening with nematocysts in real time, we can use this system to actually watch what's happening live in some of these animals. So first I want to show you what's happening with nematocysts. This is a slightly later stage animal. The cerus is pointing up in this image. The cnidosac is here, digestive gland is down here. I'm hoping you can already see these nematocysts, which are moving around in the digestive gland and can move up into the cnidosac and back out actually quite easily. So there seems to be a real ease of movement between these two structures in ways that we might not necessarily have appreciated without having this sort of imaging capability. So the question then becomes, can only nematocysts do this, or is it possible that other structures are able to move with this much ease into the cnidosac? And the answer is yes. So dinoflagellates are very easy to see in this live imaging as well. And the cnidosac up here, you can see these dinoflagellates moving in and out from the digestive gland into the cnidosac and back out again, suggesting that essentially cnidophages are interacting with a number of other tissues, not just with nematocysts. And for us, this suggests that our sort of first hypothesis, where the cnidophages have receptors to target nematocysts, is most likely the correct one, where those cnidophages have some complement of receptors that most likely allow for them to exclusively take up nematocysts and exclude everything else. So once we have this idea or understanding of where it's happening, the next question essentially becomes, how exactly is it happening? And as I mentioned, we hypothesize that it's a modification of phagocytosis receptors. This can mean a number of different things. It could mean that they've evolved sort of a diversification of receptors that allow them to target certain nematocysts. It could mean that it's just sort of a narrowing of the receptors that are available to other cell types. It's unclear exactly what could happen, but we first wanted to essentially go in and see, are there any receptors that seem like they might be relevant for sequestration within the cnidosac or within that part of the cerus? So to try and answer this question, some undergraduates in my former PI's laboratory course collected gene expression data from the distal and proximal cerus. So the distal cerus, again, is where the cnidosac is, and the proximal cerus is essentially everything else, the digestive gland, all the rest of the cerus tissue. And we performed differential expression analysis to see what genes are upregulated in the distal cerus that might be relevant for sequestration. So my undergrad, Robin Rio, performed this analysis and identified around 166 or so upregulated genes or upregulated transcripts that seem to be highly expressed in the distal cerus and may be relevant for nematocyst sequestration. Some of these genes include some possible phagocytosis genes. These are all annotations based on blast hits, so I think it's all, sort of grain of salt. But there's some really interesting ones still. including collagen alpha 5, cathepsin L, some lectin receptors, including C-type lectin macrophage mannose receptors, which I'll talk about in a second. And then some other interesting genes that don't necessarily seem to have any relevance to phagocytosis, like Wnt9, some ion transporters, G protein coupled receptors, and then gigasin, which is actually a settlement protein for chemoreception in some mollusks. I'm still trying to figure that one out a bit. But as I mentioned, if we're thinking about receptors, some interesting possible receptors are the C-type lectin macrophage mannose receptors, because we know that at least in other organisms, these types of genes are used for targeting bacteria and viruses in professional phagocytes, things like macrophages. And so they could be extremely useful for targeting nematocysts potentially, assuming they're able to target similar things on the nematocyst itself. And so I had another student, Rose Fiorenza, do some HCR in the system. I managed to get it working, and we got a lot of genes to try and figure out where they're being expressed in these animals. And we find that the C-type lectin macrophage mannose receptors are expressed where we expect targeting is occurring. So let me show you what's going on in this image. On the left here is the juvenile, the whole animal. The head of the animal is at the top of the image, the tail is at the bottom. The serrata buds are these 4 sort of little buds coming off of the side. And inside each of these is a cnidosac. And if we look at expression within the serrata, hopefully you can see the seris here, and then the green expression down here on the right, that's the expression of this gene inside of the cnidosac. So I've added the circle here to show you essentially where the cnidosac is. And you may notice it's actually more at the proximal end of the cnidosac, and this is where we expect the actual targeting is occurring, because that's where the nematocysts are entering the cnidosac structure. So it doesn't say necessarily anything about its specific function, but I think it's really good evidence that it has some relevance to what's happening early on in those sort of proximal stages of the cnidophage cells. And it brings up some questions about how other phagocytosis markers are changing in cnidophages, whether we see similar types of phagocytosis markers being expressed in similar cells, or if they're expressed in other parts of the cnidosac. We're still working on a lot of that data, but I do have at least one other expression profile to show you. So that's this collagen type 8 alpha 1 chain. Again, all blast annotations, so grain of salt. But this particular gene can be useful for conferring tensile strength on membranes. And if we think about what's happening when nematocysts are sequestered, those cells have to take up these actually quite large structures. And so it's possible that this type of gene might be really useful for helping the cell sort of elongate and stretch to accommodate structures like nematocysts when they're being sequestered. One interesting thing we noted in looking at these 2 genes is if we compare collagen 8 alpha 1 with the C-type lectin expression here, if you look inside of these circles, I'm hoping it's obvious that the expression patterns are actually a little bit different. Where we have in this gene, it's more distal end of the cnidosac expression, and this gene is more proximal end. And so this suggests there's actually a difference in expression across, not just in the cnidosac with the cnidophages themselves, but across the sort of almost development of this cell type as nematocysts are sequestered and move distally in the cnidosac. And if we look at what's happening in an adult cnidosac, it might be more obvious as to why that is. So this is a histological section of a Bergia cnidosac from an adult. You can see an adult animal on the left. The cnidosac structure is here. It has a really thick musculature compared to the other image that I showed you. But essentially what happens along the cnidosac from proximal to distal is that down here we have proliferation of cnidophage cells, targeting of nematocysts, and then sort of as they move distally along this epithelium, you get more and more nematocysts being sequestered. By the time they get up to sort of the distal end, they are essentially only nematocyst holders. They're trying to maintain those structures and potentially not digest them. And so it's not surprising when we look at it this way that there's this sort of difference in expression as we go across this structure. One last thing I want to just mention is that in order to try and get at this question, to try and understand what's happening across space, I'm also working at MBL. I'm currently here at MBL actually right now, the Marine Biological Labs in Woods Hole. to try and get spatial transcriptomics working in the system. And so my colleague or collaborator, Kate Rawlinson, is interested in Biomphalaria glabrata because she's interested in schistosomiasis and the parasite and its development. And so we're trying to get these spatial transcriptomics techniques working in gastropods, essentially, in Bergia and Biomphalaria. For me, my goal is to have essentially where we can see the histological section on a particular slide and map back expression to certain areas across the cnidosac so that we can see how that expression is changing across that structure. So that's kind of the goal of this particular work. It's still early days for this one, but I wanted to mention it in case anyone is doing anything similar and has questions or thoughts, certainly. And then, but I want to kind of end on this, how other phagocytosis markers change in cnidophages, because I think this is a really big part of this question, and understanding the molecular mechanisms of sequestration. Phagocytosis is an incredibly important part of this. And so I think for me, moving forward, this is going to be the main question that I'm targeting in understanding how nematocyst sequestration evolved, thinking about the molecular level. But I also want to point out that my work is more broad. So I've worked in, most of my background is in phylogenetics and systematics, but I've also done work in sort of ecological evolution, kind of morphological evolution, thinking about the cell types involved. And I've now started working in sort of the molecular mechanisms of this ability. And the goal of my research program in general is to get a more holistic view of how phenotypes like nematocyst sequestration can evolve. And so there's a lot of different questions that I'm interested in asking more broadly. And I'm curious about what you all kind of get out of these sorts of questions and systems as well. So to end, I just wanted to thank all of the people that have helped me with this work. None of us do our work in a vacuum, and I think it's really important to acknowledge that it takes really a village or a very large village to get some of this research done. So I appreciate all of the help I've received, including from funding sources. And I wanted to thank you all for your time. I'm happy to take any questions.
Thank you, Jessica. I will clap analogically. That was awesome. I have a question to start with, so people can either raise their hand or speak up, or if you're shy, you can type a question in the chat. This is not a question about your slugs, but it came to mind after reading the review paper you wrote a few years ago on this phenomenon in other taxa as well, where the comb jellies in the genus Hekilia also seem to be able to do some of this. And there you wrote that in Hekilia rubra, you find nematocysts around the eggs of the animal, which then the larvae eat. Has anybody looked at whether the nudibranchs, if you say there's 600 or so species that can sequester them, whether they might do something more with them than defend themselves, maybe also their brood?
I mean, we've got a number of histological sections, and I have collaborators that have looked at sort of histology of whole animals in this group. And I don't think anyone's noticed anything quite like that. And there are hypotheses as to sort of the origins of sequestration. Some people hypothesize that cnidosacs sort of originated as just a place for them to move these potentially dangerous structures sort of out to the edges, which is certainly possible. It's hard to get at that particular question, but I don't think we've noticed any other particular functions along the lines of what happens in Hekelia.
Doesn't mean that it's impossible, but that there might still be a frontier.
Yeah, it's definitely. I never say anything is impossible because we always find new stuff, but we haven't noticed anything quite like that.
Cool. I'm going to read you a question from the chat. Harriet Jameson asks, as nematocysts are mechanically triggered, how does the sea slug sequester them without triggering them or staining themselves?
This is a common question I get, actually. It's a really good question. So there's a few different ways that we know they manage this situation. One is that they have mucus that either potentially prevents them from firing, the nematocysts from firing in some of the cnidarians. That's been shown in at least a couple of species to be true. That mucus can also protect them, even if nematocysts do fire. But nematocysts do fire. So in the sort of early stages of their digestive system, they have cuticles lining their sort of oral tube and their gut as a means of protection from these nematocysts that do fire. What has been hypothesized to be happening is that in reality, that some of these nematocysts that are passing through the digestive system are not technically functionally mature. And so those nematocysts don't fire in the digestive system because they're not functionally mature. And then they make it into the cnidosac and somehow mature in the cnidosac, inside of this sort of totally different organism, which is a really interesting possibility. We have some evidence that this might be happening due to sort of acidification, which actually would make sense if you think of what happens in intracellular digestion, which is acidification essentially to break up tissues. But we don't have like a lot of really great evidence to show like exactly that that's what's happening. Yeah.
I was wondering when you showed the video of the juvenile Bergia and the little nematocysts moving in and out of the cnidosac, on the outside, on the distal tip of the cnidosac, there seem to be some sort of stiff hair-like projections. Are they involved in perhaps triggering, or what are they?
Yeah, so they have some cilia. They have a large tufts of cilia towards the tip of their serrata. This seems to be, there's not a lot of research on this, but this seems to be useful for them figuring out whether or not they should squeeze out the nematocysts. So they don't necessarily cause them to fire per se, but with their muscular part of their sac, they're able to kind of squeeze the nematocysts out. And that sometimes triggers that. Their serrata are very sensitive though. So if you kind of touch them and their serrata will orient towards whatever is touching them. So it's mostly sensory in nature, but not necessarily for firing.
It's such a cool system. I'm so happy that you're doing this stuff. So another question by Maria Sakskova. She says, is knockout available in nudibranchs? Because it would be interesting to remove the lectin receptor. I think I can guess the answer, but
yeah, that's my dream. Not yet. There are people working on this. So my former PI, Dee Lyons, is trying to get knockouts working in the system. It's been a real challenge. They're very difficult to like micro inject. We're trying to find other means of knocking out genes. I am familiar with like remote control techniques. I think they could potentially be interesting for trying in Bergia, but as of right now, it's still in progress. But I totally agree. That is like a very obvious next step, is to really knock these out and see what's happening.
I think the question by Nazit Iksi, is gene editing possible in the model organisms you used? I think that's part of your dream. And yes.
Yeah, it's exactly. It's all the same. And one way I'm thinking of trying to get around this though is there are beads that you can wash in certain proteins, and so we might be able to start thinking about what the receptors are doing if we start looking at the proteins that are in nematocysts and sort of matching those to actual uptake in the nitophages. So this is kind of, it's not exactly the same question, but it's a means of trying to at least get at what they're targeting in the nematocyst itself.
Any other questions, people? Or comments? I was just wondering if you have such a dynamic system where the nematocysts can move in and out of the cnidosac, when they get to the terminal bit, at some point, I mean, is it always that dynamic, or at some point will they be fixed and sit there for a certain number of days or whatever, and then they get resolved? Is there a sort of cycling regularity, or?
Yeah, so once they're taken up, they're in the cnidosac. They stay there. But the cnidosac it's not a static structure. I think it's easy to kind of imagine it that way because we mostly just have histological sections, but it really expands and contracts does the digestive system. Sort of when they eat, there's a lot of movement, and so it's definitely possible that they can get too full eventually. Like they might hit a point at which they have enough nematocysts. But there's some evidence that they don't necessarily store them forever. Okay. So it's unclear, but it sort of seems to be different in different species as well.
So, I see there was a hand up by someone called Manabu. I don't know if you want to speak.
Yes. Can you hear me?
Yep.
Yeah.
Thank you. Nice, great talk. I have a by the way, the movie of the cnidocytes going up and down in the nidocyst was very fun to watch, and I realized that the nidocyst has an orientation, and that should have to be aligned in the correct position. So, and the lectin recognizes sugar chains, glyco chains. So, have you noticed that? Have you stayed histologically did perform the staining that can stain glyco chains specifically? And if you see some differences or trend of staining in the nidocyte, that may give you a hint what the lectin is actually, what kind of sugar chain type that your lectin is, the lectin specifically expressed in the nidocyte is recognizing.
You mean like what on the Cnidarian nematocyst in particular? Like what type of, yeah. I haven't done any of that yet. It's definitely on my list of things. I'm hoping to bring in some Cnidarian colleagues because it's not really what I study. I think it's one of the challenges of what I do. But yeah, it's absolutely, that'd be a really interesting start because there are a few people have done work on what types of sort of structures or like proteoglycans, for example, make up nematocyst capsules. And so we can start there with our sort of hypotheses about what it could be. But yeah, the histological staining is a good idea, at least to kind of get a sort of preliminary idea about what's on the capsule and available. I think most of that work has been done in other systems like Nematocella or even like Hydra, a totally different cnidarian. And so we don't necessarily know what's happening in Aiptasia. And so I think it's possible that it's even different across cnidarian groups as well. But yeah, not yet, but hopefully.
Yeah, and the similar topic, but visualizing 2D image of what kind of material molecule are there in the tissue. You can do imaging mass spectrometry on the nematocyst to identify like the basal part, what kind of proteins or glycoproteins are expressed localized in the bottom part of the nematocyst.
Cool. Yeah, that's not my field. So yeah, maybe I'll reach out and ask you questions about that. That would be super cool. I would love to sort of supplement that with all of these other experiments for sure.
Thanks for the questions, Malibu. I think your dreams of the future, they are large, and there's a lot of work to do.
Yeah, if anyone wants to help, let me know.
Since we don't want to put you to too much of a cross-examination, another cnidarian worker, Maria Sachkova, has the final question. One more question: how do nudibranchs use the nematocysts? How is the venom released into predators?
Yeah, this is another question we don't really know the answer to. We have more hypotheses than anything else. Because they don't have a cnidocyl, right? The cnidarians use nematocysts in the sense where they have a sensory cilia on the top of the cell that are on the sort of external part of the cell that allows them to fire the nematocysts. But the nematocysts don't come with that. They still fire though. So it's possible that it's strictly mechanical. So for example, we kind of hypothesize that maybe if a fish were to take a bite of cerata, which actually regenerate, that they take a bite of the cerata and they just get a mouthful of nematocysts that fire sort of as part of that mechanical process. There is some evidence that they will orient the cerata towards like a predator or something that's bothering them, and they will sort of squeeze the nematocysts out, but they don't necessarily cause them to fire. But that change in sort of chemistry moving from the inside of the cell to outside, that can cause firing potentially. But these are mostly just hypotheses. We don't have clear evidence for any of this at this moment. We also don't know necessarily what eats the nudibranchs. So lots of questions.
We
Yeah.
need more people like you, Jessica, who start to build other species into model systems and look at the diversity of all these questions. So thank you, Marie, for that question. Thank you, Jessica,
Yeah.
for giving a wonderful talk and for being patient with the questions. All of you, thanks for joining as well. If you have a desire to give a talk yourself, or you have some friends who are doing cool stuff at the moment, just drop me an email. We are looking for talks for next month and for December as well. So with that, I would like to close the meeting. Thanks very much. It's nice to meet you,
everyone.
Italy. And I wish you a good rest of the day, everybody. And see you next time.
Thanks for the invite.
You're very welcome. Bye.